I can introduce myself. I'm Julia Schregel. I'm German living in northern Norway on 70 degrees north, so north of the Arctic cycle, and we have already midnight sun up here, although it very much looks like winter still outside. I'm a population genetist working with brown bears, and I studied during my PhD how brown bears in Northern Europe are in contact with each other, how much they travel across the landscape, and basically what the status was of the brown bears in Northern Europe in order to help preserving them and helping them to continue their existence, that we will have brown bears in the future as well. Today, the talk or this webinar is it's supposed to be like an introductory seminar that gives you some background information for the other webinars I am hosting and my colleague Alexander Kopacz is hosting, mainly about brown bears in Northern Europe. So, the first webinar is about DNA and how we can use it in science. So the content is going to be a short introduction to DNA, where it is located, its molecular structure and its function. Then I'm talking about the PCR principle. The PCR is a very important tool in genetic research. And then finally, I'm giving a short overview about what we can learn from DNA, what we can use it for. Okay, so off with the start. Our bodies are constructed of cells. And we have fat cells, and we have hair root cells, and we have intestinal cells in our intestines, nerve cells, skin cells, muscle cells, all kinds of cells. And what these cells have in common, you can see that here with red encircled with red here. You can see this little violet dot. This is the cell nucleus. This is present in all living cells in our bodies, or the bodies of every living organism, really. And inside this cell nucleus is something that we call DNA. So this is a typical cell of eukaryotes. That's a specific part of living organisms. So it's basically everything from tigers to walruses to all animals, really. So we have here all kinds of things that I'm just going to name, but we won't be really learning about those today. So you have the cell membrane that contains everything that's in the cell. Everything is swimming in cytoplasm. So a bit of a liquid buffer. We have lysosomes, vacuoles, mitochondrium. These are kind of important because they provide the energy for the cell. Golgi complex, endoplasmatic reticulum, that is called. But you don't have to remember all of this because this is not subject of our talk. What is subject of this webinar is inside here in the nucleus. And you can see this coming out here. This is the long strand of DNA. And every living cell contains one copy, at least, of this DNA. You can already see this is the chemical structure of DNA. DNA consists of 4 nucleotides. This is adenine, cytosine, thymine, and guanine, or in short, A, C, T, and G. And these 4 nucleotides fit together like so, that adenine fits together with thymine, and guanine fits together with cytosine. So adenine, or A, can never connect to C, and T can never connect to G. These nucleotides are strung up and they form kind of a ladder. You can see this guy here climbing the ladder with the rails consisting of sugar and phosphor. And here between the nucleotides is something that's called hydrogen bond. And they're like steps in the ladder. This is what is here. And you can see that along this long ladder of DNA, you have something like a sequence of letters, A, G, T, C, and so on. And this is along the whole line of the ladder, this is continuing. And this is actually where the information lies. Yes, this DNA is very, very, very tiny. And it's coiled up to a tight string, tight coils, so this whole ladder wraps around itself. And if you were to uncoil it and put one end of the DNA ladder to the beginning of the next end, the next DNA ladder, and if you were to do that with all of the DNA that is contained in one of our bodies, so one human, the DNA of one human is so long that it can cross the diameter of our solar system 2 times. Isn't that amazing? I think that is something that strikes me always as almost unbelievable, that we have so long DNA in our bodies that we can go from the sun to Pluto and back several times over. And that is truly remarkable. So you can imagine there's a lot of information stored on this DNA. You can see here this string of letters. And what is actually going on here? What is this information? So we come to the function of the DNA, the principle of the DNA. The first one is replication. So from one string of DNA, we have 2 strings of DNA afterwards. And this always happens, of course, when our bodies grow, when the cells divide, when from one cell there will be 2 cells. And what is happening here is that a certain enzyme, it's called DNA polymerase, travels along this string of DNA, this double helix, and unwraps it, and then building blocks of the DNA, you can see them here, they are called dNTPs. Here is the nucleotide, T, A, and G, and then there's this phosphor group, a chemical compound attached to it, and they kind of swim around in the nucleus, and when the DNA is about to replicate, they come swimming along to the DNA polymerase, and the DNA polymerase travels along one string, like for example down here, and then it grabs these dNTPs that fly around and attaches them in the appropriate place. And you can see A, T, G, C. So here's the new string of DNA, and here's the, like the blueprint. So here on this place, there would be this C nucleotide, the CNTP attached to it here. And then this one would be a G, and then would be here an a T attached to it, here an A. So you have the complementary information on the one hand side of the DNA. And this travels here in this direction, and this kind of shoots out here in the end. And so in the end, you have 2 copies, 2 identical copies of the DNA. This is one of the primary functions, or the things that processes that going on in the cell nucleus. The next one, and here we come to the biological function, is to build proteins. So a protein is a long string of amino acids, and they are one of the key actors in our bodies. They help for the functioning of biochemical reactions. For example, how our energy is consumed in our body. the food is processed and so on. That all takes energy, and proteins help with these reactions. They are responsible for cell signaling. If the cell signals we need more water and all this kind of stuff. Immune response, if we get sick, then proteins are responsible for saying how our bodies are supposed to react to this. So the white blood cells, for example, how they are supposed to attack the virus or the bacteria, and also structural function. So our muscles and our fat cells, how they are built up and how our bodies look like. This is all regulated and done by proteins. And you can see here already, this here is the primary structure of a protein. So you have a string of amino acids. Here every bead is one amino acid, and there are several amino acids. I'm not going to go into the details of this because this is supposed to be just an overview. And they are kind of like a pearl string attached to each other. And if you let go of the string, it first curls up here like so, like a little spiral. And then you have this spiral or the string, this kind of, weirdly forms, like a mess of a long coil. And these individual proteins, they can form large molecules consistent of several proteins. So this is quite complicated. There's a lot of research going on how these proteins are actually folded and what makes them fold this way. So, but for now, we just focus on the fact that DNA codes for proteins. How does it do this? Well, there's 3 parts of the protein biosynthesis, it's called. So the biosynthesis just describes that it's a natural process. It's nothing artificial about it. You can also replicate it artificially, but this is going on in the cells of our bodies. So you need 3 things. You need something that's called mRNA or messenger RNA. You can see this looks kind of familiar, doesn't it? It looks like the DNA. The difference is that it's only one string. So it's not a double helix, it's just one string. And, you can't see it here just as well because it's a little bit small, but you have also 4 nucleotides. You have cytosine, the C, you have guanine, the G, and you have adenine, the A. But instead of the thymine, the T from the DNA, you have something called uracil, and U. This is not so important, but that is one of the differences between DNA and RNA. Then you have the second part is the tRNA or the transfer RNA. This is kind of a neat thing. You can see it's also RNA, so it's essentially also a string of nucleotides, but they're kind of wrapped up here and form like a little T almost. On the one end is an amino acid attached, and here on this end is the anticodon region. I'm going to come to that in the next slide, what this is. And then you have the ribosome. You may remember that I'm going back to show you. Do I have this here? Yeah, you have this called endoplasmatic reticulum, and the ribosomes are attached here to this endoplasmatic reticulum. It sits on top of this here, this yellow structure. So you have ribosomes. So these 3 things are basically the workers of the protein biosynthesis. How this works? Okay, we have the DNA in the nucleus of the cell. This is the DNA. Here it's still coiled up as a double helix. The first part is called transcription. So you remember you have a string of letters, really, the DNA, A, D, G, T, A, and so on. It's a long string of letters. And we have this RNA polymerase that is a little bit, or that is similar to the DNA polymerase. So it's an enzyme that unzips the DNA at a certain part that it wants to read and then travels along the DNA and then builds up a piece of RNA, a string of letters, basically, the messenger RNA. And this messenger RNA has the unique feature that it can travel out of the nucleus into the cell, into the cytoplasm. And then you can here, I try to show it here. This is the string of letters, C, G, U, A, U, C, G, U, C, just jotted along something that doesn't necessarily have a meaning what is standing here. And you can see here, this is the tRNA. Here is the anticodon region. So you'll see that this here attaches here, right? So we have here on the messenger RNA, CUU, and here the anticodon region, GAA. So all these tRNAs have a specific anticodon here. So the information contained in the DNA consists of the sequence of 3 letters, really. And the sequence of these 3 letters, or these 3-letter combinations, are called codons because they code for a specific amino acid. Here, for example, AAG codes for an amino acid called lysine, and is attached here. And CGC, that is the one that attaches here, has, I think it's called arginine. I'm not 100% sure because I'm not a my main focus of research is not on protein biosynthesis, but I looked it up in a table. So this is, I think, arginine that is coded with this here. So every 3-letter code codes for one specific amino acid. Sometimes different codes code for the same amino acid. So not all combinations lead to a different letter combinations lead to a different amino acid. But yeah, that would be going too much into the details. So the tRNA comes flying here into the ribosome. The ribosome attaches itself here to the RNA, travels along, just like here the RNA polymerase does, travels along here the messenger RNA string and fetches tRNA that comes flying here, and the proper tRNA attaches to the proper place here, and then the amino acid that it carries on its tail is attached to the ones that are already there. So this is already amino acids that have been used to construct a string of pearls. And the amino acid chain comes out here, and the now empty transfer RNA can fly and fetch another amino acid from the cells. That's why it's also so important if you hear people talking that you should eat, for example, I don't know, linseeds, for example, or certain types of vegetables or so on, because there are important proteins and amino acids in this food. This is the reason for it, because we cannot produce ourselves, I think, most of these amino acids, so we need to get them somewhere, namely from our food, to be able to produce the proteins that our body consists of. These are the 2 parts of the protein biosynthesis, the transcription and the translation part. So first the messenger RNA comes out of the nucleus, and then this is the messenger RNA is basically the blueprint that is translated from a string of letters into a string of amino acids that form the proteins of our bodies. So the DNA consists of so-called coding regions. So we have, like I just told, regions on the DNA where the sequence of letters code for certain amino acids, and they're used to synthesize or build proteins. But then, weirdly enough, there are also non-coding regions. That means all of the regions which apparently do not code for a protein. Some of these regions are responsible, for example, how the RNA is built, because all of these, the tRNA and the ribosome, well, the ribosome is also a protein, but the tRNA, the messenger RNA, this all needs also, of course, be encoded here, because how else is the cells are supposed to know how to build a messenger RNA or a transfer RNA. So this information is also contained in the DNA. But then there are also non-coding regions, and lots of it, on our DNA, where up to now nobody is really know what is their biological functions. They do not seem to code for a certain protein. They do not seem to code for messenger or transfer RNA. They don't seem to have any regulatory functions. So basically, there's a little bit unflattering term that was made for it, is junk DNA, because it's just sitting there without any apparent region, without any apparent function. There are some theories or some hypotheses to why it's still there. Some say that it's leftovers from our evolution. So at some point they had a function, but since during evolution they just lost the function. There were maybe some mistakes in the process of replicating the DNA, and they just lost the information. They don't have a biological function, so they are not purged from the DNA. It's just the DNA doesn't bother to get rid of it. But there's still a lot that we do not know about this so-called junk DNA. So there are many researchers who do not like the term junk DNA. And now we come to what is important for the next webinar that I'm hosting. It's called DNA markers. So what we do in genetic research is that we look at specific parts of the DNA. The markers are basically variations in the DNA sequence among individuals and species. There are variations in the coding regions, which may alter amino acid sequences. So I go back here. I should have had this here, but I didn't put it there. If you imagine that, for example, instead of the UCC that stands here, there has been made a mistake here in the messenger RNA synthesis, and instead of a CUU, suddenly we have CUG standing here. Then this could not attach, the transfer RNA could not attach here because it's the wrong code. And then instead of the lysine following the arginine, we maybe had another amino acid following this arginine. And then the structure of our protein would be altered. And that may be quite important for that organism. For example, I guess many of you have heard of the term sickle cell anemia, where the red blood cells are formed differently, abnormally formed, and they cannot carry oxygen because of that. And this sickle cell anemia is due to a mutation in the coding region for a specific protein that is involved in building up the red blood cells. So there is the wrong string of amino acids, and the protein just comes out wrong. And because of that, the persons who have sickle cell anemia cannot do not have as much oxygen in their bodies. So you have variations in the coding regions. It may not always be bad for you, like with the sickle cell anemia. It may also be good that there is a protein that functions much better than another one because of a variation in this coding region. But you have also variations in non-coding regions. And these variations often have no known biological consequence. So they are there and we can see them, but we don't know of any function. Two of the markers that I have used in my research are called short tandem repeats and single nucleotide polymorphisms, or STRs, or microsatellites, or SNPs. Here you see the long string of the DNA again. And here you see something peculiar. Here you have the normal code, A, G, C, T, G, C, T, and suddenly you have this T, A, T, A, T, A, T, A, T, A, T. So it's basically like the DNA has gotten a hiccup or something. So you have the same sequence of letters that repeating itself over and over again. And this, we have quite a lot of places like this in our DNA. That's why they're called short tandem repeats, because this is a tandem, this AT, and they are just repeating itself. Sometimes it can be just two letters, like this TATA, but it can also be three, like I don't know, GCT, GCT, GCT all over again. These variations carry so far have no known biological consequences, so they just sit there, but we don't know if they do anything. And then we have SNPs. If you look closely, you can see that these two sets of double helix, of DNA double helix, looks similar, apart from this region here, where this is guanine and cytosine, so blue and yellow. And this here, on the other set of DNA, instead there is an adenine and a thymine, so green and red. You can see here says what the colors stand for. So these two sets, or these two strings of DNA, are identical apart from this one base pair or nucleotide pair here. Otherwise they are identical. And these types of places, we have a lot of them in our DNA, where everything is identical apart from one pair of nucleotides. And these kind of variations can be in coding and non-coding regions, with the consequences that I already told. So sometimes a SNP codes for different amino acids, and sometimes it's in a non-coding region, so it doesn't have any apparent biological function. And these two markers we utilize most, or I utilize most in my research. If we want to look at the DNA, I told you it's very, very tiny. If it can wrap 2 times about our around our solar system, you can imagine how tiny the DNA must be in our cells. And it was quite problematic for a long time. How do we actually manage to look at it? And then came a smart man called Mullis, who figured out something that he called polymerase chain reaction, or PCR in short. I've posted a short description also on this Polarpedia. And basically what this polymerase chain reaction does is imitating what the cells do all on their own when they are replicating DNA. So it produces a replicate of the DNA with a sequence of distinct temperature changes. So when you heat up DNA, this is where we start. I'm going through this step by step. If you heat up the DNA, this double helix, to 95 degrees, what happens is that the hydrogen bonds that form the steps of the DNA, they break. And so this double helix becomes two individual strings of DNA. I try to visualize it here like this. So they kind of move away from each other. Then in the next step, the DNA is cooled down again to 55 degrees. And then we have something here that attaches itself. This is the marker that we are attaching. It's basically a string also of nucleotides that is complementary to the DNA here, and they put themselves here. You put them, you put them in the chemical reaction that you're building. You put them together with the DNA, and they put themselves towards the appropriate region on the DNA. And then you're heating the whole mixture up again to 75 degrees. This is all automated, so you're not standing there on the stove and doing that. So you're heating them up to 75 degrees, and then come this polymerase, this artificial polymerase comes, attaches itself to this region here, it recognizes it, and then it starts building, like what's happening in the cells with the DNA polymerase, it starts building a second set of DNA string, so the other part of the ladder here. And it starts here at the marker where we put the marker. So it starts here and then builds down here, and then this one on this other side, it builds into this direction. So this is cycle 1. So you see already we started out with 1 copy of DNA, and now we have 2 copies of DNA that are slightly variable because here there is nothing there, and here's nothing there. And then we have cycle 2. So again, the DNA is heated up to 95 degrees. So this double helix detaches itself from itself. The hydrogen bonds break, and you have 4 individual strings of DNA. The marker DNA comes, the string of nucleotide comes and attaches itself to the certain part in the DNA here and here and here. Then we heat again, but to 75 degrees, and then the polymerase comes, attaches itself to the marker, and travels along the string of the DNA here and builds up a replicate. And then in the end, here, just to remember, it goes here in this direction and down here and up here. And then we have something after cycle 2 that looks like this. So we have the original DNA that is still here. And then we have short bits of DNA here. And this is the target sequence. That is what we want to look at. I will go back a few slides to show you. You can see here, this is if I wanted to look at a microsatellite, for example, this year, then a marker sequence could be, for example, here, this AGCT. That is a sequence that is, this is where I attach this orange marker that was in the, what I showed you here in this slide. So the marker sequence attaches here, for example, and then it travels along here and builds up the DNA along this string, and this happens also on the other side. So here on this other one, there's also a sequence sitting here, so the marker sits itself here, and then the DNA is replicated here along this string. And this is also the same that's happening with the SNP. So the marker maybe, if this unravels, then the marker attaches itself here and then builds along one of the strings of the DNA. I know it's very complicated. I'm going to repeat this a little bit in the next lesson also to remind you of this. So this is hopefully not the last time you hear of it, but this is a fantastic process where in the end you have typical cycle. You have usually what I use in my research, we have, for example, 25 cycles. And because you're always doubling, in every cycle you're doubling the amount of DNA, you end up with a huge amount of DNA. And it's so much DNA that with special machines, this comes in the next webinar, you can actually visualize it and you can see the signal of this DNA and look at the variation, understand where the variations are. So you can compare the code or the string of letters of the DNA among individuals, for example. Okay, what was the next? Okay, so what can we learn from the DNA? You saw that the process of replicating the DNA with the polymerase, I go back there again, with the polymerase here building and replicating the DNA, you can easily imagine that sometimes, not very often, but sometimes there is a mistake happening, that instead of a C that is supposed to attach here, there happens something that instead a G is attached or an A is attached. It can happen, for example, with just because of these mistakes in the replication process, or in the when something is broken and the DNA gets repaired, that suddenly this code is altered. And also it's called mutagenic forces. For example, UV light. You've all heard that you're supposed to not go in the sun without sunscreen too often, because if you get a sunburn, that is actually what's happening, that the UV light knocks out or destroys parts of the DNA. And then sometimes that is not repaired properly again. So the DNA in the cell is then altered, and this may lead to, for example, cancer cells forming, so or damaged DNA. Not all of the mutations are bad, like I said. Sometimes they turns out that the altered proteins perform better than the original protein. So if you have this going on over a long period of time and over the course of evolution, you may end up with a different organisms may change their appearance slightly. They may have different abilities. That, for example, the muscles of built with a altered protein are stronger and can perform better than with the original protein. So suddenly, or not suddenly, but this organism performs slightly better. Maybe it can run faster than individuals with the original protein. And tolerance, maybe, for example, changes in the DNA lead to that DNA can be better repaired. So we have a higher tolerance for UV light, for example. And all this leads to increasing speciation. So we have an increase of variation among all the organisms on our planet. And this is traceable by comparing the DNA of different species and different genomes. So the genome is the entirety of all the DNA. And then we can have this tree of life. Individuals inherit DNA from their parents, and DNA is unique to everyone, so we can identify individuals and we can look where they come from. So we can, for example, find out maybe is Prince Philip related to the Russian Tsar. We can look at where wildlife crime, for example, and population structure, where individuals come from. This is from bears in Finland. Do the bears come from Russia, or are they original bears from Finland? And we can find out if a bear was shot in Sweden, if that maybe was a Norwegian bear that was identified in where I come from, that has traveled to Sweden. Yeah. And for example, if there was a disaster and there many people have died, we can use DNA to identify them, who it was actually that died at this specific spot. Yes. So I'm noticing that I'm a bit over time. So if there are questions, just question me, or pose your questions now, and I'm trying to answer them.